All right, team. So this week we are talking about animals and what makes an animal, right? Well, animals are multicellular eukaryotic organisms. They are organisms that are heterotrophic, meaning that they have to eat something else or get their nutrition by consuming other organisms. This can be either herbivorous, carnivorous, omnivorous, they can be parasites, right? But they have to eat something else to get their food, to get their energy. And animals are motile. They can move around at least at one stage in their life cycle minimum. So even if they're maybe sessile as adults, maybe their larvae are free swimming or free moving, right? But this mobility, this ability to move, is only possible because we have specialized tissues. Animals have specialized tissues, nervous and muscle tissue, which allow for that movement. Okay, so really at the core of it, that's all an animal is. They're multicellular eukaryotic organisms with no cell wall. They're heterotrophic, having to eat something else to get their food, and then they can move. And they move because they have nervous and muscle tissue. So from here, we're going to talk about some of the general characteristics that we use to break animals up into different phyla and what those terms mean. Okay, so one of the big questions that we ask when we're talking about animals is what type of symmetry does their body plan have? So all animals develop from the same basic set of genes that control body plan, but variations on that genes and their copy number and things like that give us many different body plans. But at the core, we have three different types of body symmetry. We have organisms like these ones right here, these sponges that would be in phylum Porifera. They are asymmetrical, meaning that they lack any sort of body symmetry. So what that means is that there's no one line that we can draw through this sponge where it would be the same on either side, where it would be a mirror image of it on either side, right? The other main type of body symmetry that we see is radial symmetry. So this is where the organism has multiple lines of symmetry that we can run through it, and it will be the same on either side. These are mostly going to be the organisms that we're going to look at are going to be the cnidarians. So things like anemones or corals or jellyfish, things like that. And then the third major body plan or type of symmetry that we see is bilateral symmetry. So bilateral symmetry, we have one line of symmetry running from the anterior region to the posterior region, head to tail. And if the organism has a tail, I guess. So we're going to be the same on the left and the right side. And this bilateral symmetry is associated with something that we call cephalization. Cephalization really just means a head. So now in bilateral organisms, we see that they have a head. And what's a head at the core of it, right? A head really at the core of it is just a concentration of nervous tissue and sensory tissue. So, right, like on this goat right here, we got the senses of smell and taste, it can eat. It's also hearing and vision, right? And it's got its brain here too, right? So bilateral body plan, it's laid out so that way you're moving in one direction and your head is typically facing in the direction in which you're moving. So that way all of your sensory organs are looking and hearing, smelling, tasting, however that organism senses its environment is in the direction of movement. So we use either asymmetrical, radially symmetrical, or bilaterally symmetrical in order to break up these organisms into different phyla. So that's one of the questions that we ask. The other question that we ask, that might sound kind of silly, but we ask, is that organism a protostome, or is it what we call a deuterostome? And what do these terms mean? Well, we've kind of seen some of these pieces before, right? So proto basically means first, and deutero means second, right? What about stome? We've seen this word before. We saw this word when we talked about plants. Stome means mouth. Okay, you're like, probably by now, where are we going with this? So let's look at what's going on in an animal really early on in its embryonic development. And that's where these terms come from. All animals develop from a single-celled fertilized egg that we call a zygote. So this is an egg that has just been fertilized by a sperm. That, in animals, this zygote, it's now diploid, right? Because the egg was haploid, sperm was haploid. This diploid zygote then goes through a process called cleavage, where it goes through cell division without growth. And then eventually, after a few rounds of that, it ends up in a hollow ball of cells that we call the blastula. So now something really unique happens in animal development called gastrulation. And that process is what sets up that protostome versus deuterostome. And that process of gastrulation also sets up our digestive system and our tissue layers. Okay, so what's gastrulation? So in gastrulation, what's happening is that some cells start to migrate and move inwards. They start to create this kind of infolding into the embryo. And this opening now, this opening that's right here, this is what we call the blastopore. Okay, so the blastopore. This is significant because in some animals, that blastopore might become the mouth. In some animals, that blastopore becomes the anus, and the mouth forms later. So this blastopore, so these cells are going to continue to kind of move inwards, kind of like that, until we get two openings for some animals. Some animals only have one opening, but this first opening, if it becomes the mouth, we say that they are protostomes. If this first opening becomes the anus and this second opening becomes the mouth, we say that they are deuterostomes, mouth second. Okay, so this layout of what's happening in the embryological stages in order to set up the mouth, and then this middle section right here, this middle section becomes the digestive tract. What's happening in the embryological development is how we determine protostome versus deuterostome, and that's one of the major differences between a lot of the animal phyla. So kind of if we were to turn this embryo on its side, kind of like a little like this. If we were to turn it on its side and kind of cut it in half the other way. So we can see we're kind of basically a tube right here. This would be our digestive tract, and then an outer layer of cells. So in some organisms, we say they are triploblastic. They're going to have 3 layers of cells. So they would have a third layer of cells in the middle. And those would arise from cells just coming up in here and continuing that infolding. So in some organisms, they have a middle layer of cells that we call the mesoderm. So we would have 3 layers. So we'd have the digestive tract layer that we call the endoderm. We have a middle layer of cells right here that we call the mesoderm, and then we would have an outer layer of cells that we call the ectoderm. Some organisms like cnidarians, though, only have an endoderm and an ectoderm. They're lacking this mesoderm. Okay, so now let's apply that to talking about our animal phyla. So we have all animals we think are from one common ancestor. Very early on, one group of animals broke off. And those are going to be the sponges, phylum Porifera. So the sponges lack any symmetry. They are not bilateral or radially symmetrical. They don't have symmetry at all. They're asymmetrical. They even don't have specialized tissues. And they're sessile as adults, but they're motile as their larval stages. The next major division that we see is the cnidarians. These organisms have 2 tissue layers, and they are radially symmetrical. Then down here on this other branch, everything last here is going to be triploblastic and bilaterally symmetrical. But we get two major divisions because one division is going to be protostome, the other division is going to be deuterostome. Okay, so this lineage right here, so let's go back and let's mark on some of these synapomorphies. So those traits that we use to categorize these organisms into different phyla. So this would be our common ancestor of all animals. Right here, at this point right here, we see specialized tissues. So that means that everything past this point in the evolutionary tree has specialized tissues. So even though cnidarians only have two tissue layers, endoderm and ectoderm, they still have specialized tissues. After this point right here in our phylogenetic tree, we see everything past this point is bilaterally symmetrical. Then, those organisms that are bilateral, we see two major divisions. The first division is protostome development. So everything on this point onwards is protostome development. The other grouping are the deuterostomes. So mouth first versus mouth second. So now let's talk about what some of these phyla are. Up here, we're going to write in the arthropods, Arthropoda. Those are things like insects and crabs and arachnids, spiders, scorpions, things like that. And the other major group over here is going to be the Nematoda. And the reason why these two are so closely related to each other is because they share the synapomorphy.
Ecdysis.
And basically what that means is that they have to shed their outer layer of skin in order to grow. So they shed their skin, they get rid of that outer layer in order to make room. And these two also have exoskeletons of a sense made out of chitin. This group down here shares the quality of having lophophore feeding structures, or what we also call another trait that they share in common is they could potentially have trochophore larvae shape. Okay, and so those organisms they're more than the phyla that we have here, but we're going to put the annelids here. So the annelids are the segmented worms, phylum Annelida. Also in this group is phylum Mollusca. So Annelida are the segmented worms, phylum Mollusca are things like cephalopods, which include squid and octopus, as well as things like clams, bivalves, slugs, and snails, things like that. And then the last group also is going to be platyhelminthes. So platyhelminthes are the flatworms. Platy, like plateau, means flat, and helminth means worms. So the platyhelminthes are the flatworms. They include things like many parasites, like tapeworms. There are also some free-living versions as well, but this group is really known for being tapeworms, or for being parasites rather. Okay, but there are many free-living species as well. And then in the deuterostomes, remember deutero meaning mouth second in the embryological development, we have two major phyla. We have the chordates, which includes us, phylum Chordata, and we have another phylum Echinodermata. So phylum Echinodermata, this includes things like sea urchins, sea stars, sea dollars, things like that. And chordates is everything from us to fish to hagfish to lampreys to everything with a backbone, as well as a few things without a backbone as well. So that's our general overview of how we break up our animal phyla and kind of a look at our phylogeny and how they're related to each other. And I hope that was helpful.